Hello, and welcome to the Xcell Biosciences webinar. We have two presenters today, Dr. James Lim, Chief Scientific Officer and Founder, and Dr. Bruce Adams, Director of R&D. After the presentations, we will be taking questions. Please type your questions in the chat box. And we thank you for attending today. We will now begin with Dr. James Lim.
Thank you, Sheila, for your kind introduction. And before I begin, I'll provide a brief overview of today's topics. I'll start by providing a high-level overview of the technology we have developed, a novel pressurized bioreactor that can essentially squeeze cells through gas compression. I can tell you a little bit about the theory of how it works, and then we'll spend the next 30 minutes or so telling you what we can do with pressure and elucidate the impact on human cells. And my colleague, Bruce Adams, will follow that up with stem cell differentiation and immune activation application data. So why don't I just dive right in. So the question that we're trying to ask here is, why pressure? What happens to cells when we put them under pressurized culture conditions? As we all know, the body is made up of many different microenvironments, all with its own unique molecular compositions, including dissolved oxygen and fluid pressures, that is often very dynamic. It's in these diverse microenvironments in which cells reside, and the cell surroundings can dictate the behavior of cells at the molecular level. Many of you are probably familiar with low oxygen conditions, but in conjunction with dissolved gas levels, interstitial fluid pressure is also an important and dynamic parameter. For example, as we breathe in air, our lung cells stretch and contract. Our heart cells beat and pump blood. And as we walk, the muscles in our legs contract and stretch. All of these are basically mechanical forces and cues or signals that a cells can sense and in turn respond to these extracellular inputs. So we typically like to cite the work of Dr. Greg Semenza at Hopkins, who discovered hypoxia-inducible factor, a master transcriptional regulator critical for the development, for neuronal development and development processes, and also implicated in disease progression. And Dr. Rakesh Jain at Mass Gen Hospital, Harvard Medical School, who has spent the better part of 3 decades researching the impact of pressure and mechanical forces on the behavior of cells. As shown in this graph, you can see that various tissues and tumors have varying oxygen and pressure levels. And in order to successfully model these diverse microenvironments, we created the Avatar Cell Culture System, an array-based culturing workflow that allows users to vary multiple oxygen and pressure conditions simultaneously. So to get straight to the point, what happens to your cells under pressurized culture conditions? And by me giving you the answers first, I hope to sort of convince you over the next 30 minutes that we have some very exciting data showing that changes could be observed in cell morphology, gene and protein expression, as well as changes in cell metabolism is observed under pressurized culture conditions. So for stem cells, when you put stem cells under pressurized conditions in conjunction with low oxygen, we'll show you that we can actually further differentiate these cells into cardiomyocytes and neuronal precursor cells. We can also show you that we can take fresh tumor biopsies from patient samples, namely carcinomas, and perform long-term propagation of primary tumor cells ex vivo. We'll also share with some data sets regarding immunotherapy applications, where we'll show you our workflow for novel target discovery and drug screening applications. So commonly asked questions regarding the avatar is, how does it work? And I'll show you that in the next slide. The other question is, how does a cell sense or feel pressure? And then finally, how do I know what pressure levels to use? So cells cultured in the Avatar bioreactor can be thought of as a hyperbaric chamber for cells. And the pressure in which the cells feel are predominantly contributed by three major components. That's the pressure exerted by the culture media, as well as a small amount of pressure level that's typically observed through the atmospheric air, and that will vary depending on whether or not you're at sea level, below sea level, or above sea level. And finally, the avatar bioreactor, where through our gas compressor, we can create a highly pressurized internal chamber. And so without going too much into the physics or the math, what we can sort of understand is that the total pressure exerted on the cells, again, is contributed by media and air pressure, as well as the avatar pressure. And this diagram is just to basically show you that the pressure forces contributed by the media as well as the ambient air is minimal compared to the total avatar pressure in which the system can model. And so just as a reference, when I give you a value of 2 psi, that means it's 2 psi above ambient. And so the absolute value is taking the 0 reading, which is 14.7 psi absolute, and then adding 2 psi on top of it to create 16.8. But all you guys have to know is that the avatar pressure created by the avatar bioreactor can create pressure levels that far exceed the contribution of media as well as the ambient air pressures. And so just to quickly summarize, the avatar bioreactor is the only system, in contrast to its competitors, conventional CO2 incubator or hypoxia workstation, that can actually simultaneously use gas-based pressure to squeeze cells. So the next question is, okay, so what happens to cells under pressure? And what measurable change can we observe at the molecular level? And so this is a PCA plot, a principal component analysis plot, showing work performed on primary human fibroblasts that were cultured under varying oxygen and pressure conditions. We have 9 total conditions in which we cultured these cells. And when we looked at differential gene expression analysis, we found that we could cluster sample sets based on whether or not pressure was applied as a function of the culture condition. And as indicated by this plot, with increasing pressure levels from 0 psi to 2 psi, you could significantly alter gene expression signatures from samples that were cultured under 0 psi, 0.5, or 2 psi. And in the same data set, we can also start to collectively group these cells based on whether or not oxygen was applied to the fundamental culture condition in question. And what this basically tells us is that we could start to look for pressure-sensitive genes and oxygen-sensitive genes. And these are critical for biomarker validation as well as target validation workflows. But so what does pressure do to cells? At a very, very high level, when we look across a spectrum of 4 different cancer cell lines, we typically find that cells under pressurized culture conditions show significant changes, both downregulation and upregulation of various metabolic processes as well as nuclease activity. In general, the 4 prostate cancer cell lines that we looked at show that there's about 100 genes that overlap between oxygen-sensitive regulated genes as compared to those pressure-regulated genes. So I showed you some data regarding gene expression changes, and I would like to finally sort of show one little piece of data before I hand off to my colleague, Bruce Adams, who will tell you a little bit about stem cells. But here we're showing a very simple experiment where we're looking at the expression of androgen receptor variants. And in this case, ARV7, variant 7, is a drug resistance marker that seems to promote or suggest rapid progression of prostate cancer. And what we show is that androgen receptor alone does not seem to be altered in terms of its protein expression as a function of the different avatar conditions in which we're modeling, both at 1% O2, 2 psi, tumor microenvironment, or ambient oxygen with pressure alone. And here what we show is that with the application of 2 psi pressure, which is similar to the arterial blood flow, as well as the pressure found in the metastatic bone marrow, we find that AR variants are increased in terms of their expression patterns compared to the wild-type androgen receptor. And what this shows is, again, a proposal for a mechanism of action to determine whether or not resistance markers are virtue expressed due to the clonal nature of the cell type, or perhaps certain markers are expressed just because these cells are in the correct microenvironment or in the aberrant microenvironment in question. And so with that, I will hand off to my colleague Bruce Adams, who will tell you a little bit about stem cell differentiation and immune activation. Bruce.
Thanks, James. All right, looking forward here to discussing a little bit some of our applications in stem and immune cell biology. Again, overall, we're trying to set the avatar in a place where it really can enhance current protocols that you already are working with in your stem cell and immune cell biology case. All right, so users have already begun applying avatar technology in various workflows. So I will speak to first looking at some of the cases for stem cell biology. Now, why stem cells? Well, increasingly biologists are using the power of iPSCs to complement their traditional cell line models and mouse models of human genetic diseases. There's a variety of major advantages to using iPSCs outlined here. The reality is there's still a number of inefficiencies in the methods and time to generate iPSCs, as well as maybe more importantly, differentiating them into mature cell types for both drug discovery work, toxicity work, and so on. Labs have increasingly looked at now as there's core facilities and labs as they're being asked to increase the number of lines they work with. They're scaling their reagents, their workflows, and they're looking for opportunities to save time and money in their workflows. And we want to show you some data where we think the avatar fits nicely in there. So some key areas users are already using avatar technology, including enhanced reprogramming efficiency, enhancing the influence of current standard induction protocols, and as well in differentiation protocol. And I'll show you some examples of that for neural differentiation workflows. But first, and to build on what James brought up, what do we know about hypoxia and physical forces in stem cell biology? Well, it turns out that hypoxia has been well studied in the context of pluripotency. Among many groups, including Yamanaka's group, showing some work here from 2009, it showed that there's increased reprogramming efficiency in low oxygen. Now, if you go too low, you begin to reduce some of this efficiency again, as shown in their model. But in this case, they show that 5% O2 yields the most number of GFP positive colonies in transgenic OCT-GFP mice embryonic fibroblasts. If you look here on the right, Mathieu and colleagues showed that hypoxia at 5% O2 can actually revert early committed human embryonic stem cells back to a pluripotent state. So embryonic and adult stem cells, they do sense pressure as well. Now you can think of this from from early rapid proliferation of ES cells in the inner cell mass of a developing blastocyst. It's actually postulated to increase pressure as the cells begin in that confined environment to divide and make contact with each other. Basically, this is a case where mechanical forces can regulate ECM integrin signaling pathways, that govern stem cell self-renewal and differentiation. In the middle here, this is work by Engler from Cell in 2006, showing how mesenchymal cells actually respond to mechanical forces to induce differentiation. In this case, they actually manipulated the substrate stiffness on which they grew their mesenchymal cells and created different lineages based on that. And on the right, wave culture method is a common way that people are expanding cells, but there's actually a shear stress component to these cells, and that shear stress is shown here to actually influence the differentiation rate of cardiomyocytes. So in our case, we have the avatar, and that allows us to control and manipulate independently pressure and oxygen. And what we show here is that you can actually increase your reprogramming efficiency under a particular pulse setting of a combination of very low oxygen and higher pressure to actually optimize of your reprogramming efficiency. In this case, the readout being the number of colonies generated per 100,000 fibroblasts. Building on this sort of early data, we've had users now apply this to look at scaling workflows and finding efficiencies in this case, in a core facility setting. At this point, they were able to use our recommended reprogramming settings in the avatar to obtain larger colonies. So at the high, which at 19.7 psi, which is atmospheric in their case, plus the 5 psi from avatar, they found above average morphology and size for these cells. Now, what this allows these users to do is actually pick cells much earlier, about a week earlier actually in their workflow, and move these cells to downstream other projects. So there is an influence in colony number. They had more uniform colonies, so the colony quality control they were looking at suggested that alkaline phosphatase was more evenly expressed across the entire colony. And the cell quality within the colonies was also seen to be enhanced in the bottom right by measure of OCT4 and TRA160 co-positive staining. So in understanding a bit more of the biology and the influence of pressure and O2 regulation on iPSC maintenance, we had a different set of experiments here. So what we were able to show here, this is just under maintenance media. If you look here at the top line, these are the images, the stainings of NANOG, DOC4, SOX2, and SSEA4 under standard culture microenvironment. On the middle row, this is under avatar when we have under a hypoxic condition, 5% O2 with no additional pressure. And then we've added a pressure component on the bottom row here. And what you can see is that we actually lose NANOG expression, and OCT4 expressions dissipates here. We also lose SSEA4 expression. We're actually able to induce differentiation of these cells just by controlling pressure in this setting. Now, qPCR analysis down here shows that confirms the loss of some of these pluripotency markers, as in NANOG reduction and OCT4 reduction, and we see enhancement of markers of early neural differentiation, as indicated by NCAM, Nestin, MAP2, and the ectodermal lineage marker PAC6. So further analysis by RNA-seq at passage 5 of these cells showed that confirmed the upregulation of neural signature. So there was multiple lines generated and tracked under these conditions in each case. And here you can see that the neural signature is enhanced under the hypoxic and pressurized condition only. We further postulated about what might be driving some of this biology, and you can see here on the right a list of top differentially expressed genes from these mRNA-seq data that would code for secreted proteins. And you can see, for example, a number of reasonable candidates such as noggin are expressed here that are known to influence this cell state. So that's the influence of pressure under a maintenance media. So can pressure enhance your normal workflow for neural induction? And I'm showing you now here that it can. So this is using a standard soluble workflow, soluble factor workflow to induce neural cells. And in this case, this is being performed on adult iPSC lines generated in-house, as well as neonatal iPSC lines. And what you can see, there is some benefit at low oxygen alone compared to a standard incubator. Overall, though, you get the most consistent expansion of colonies and induction of colonies under both hypoxia and pressurized conditions. In this case, you can see here the ratio of PAC6 positive cells is significantly increased in adult and neonatal iPSC lines under these conditions. So what about the ability to further downstream? Now, if we move into differentiation protocols, can we influence cells further there using the avatar? In this case, we were looking at a motor neuron differentiation protocol, and this protocol was based on one published protocol by Mori et al. From 2015. Showing you here, looking at markers for at day 10 to look at OLIG2 expression, and what you can see already at day 10 is that we have enhanced OLIG2 expression under the avatar defined settings. And by day 31 of these cultures, you can see by these images, we have clearly enhanced an earlier expression of neurofilament H and cholinesterase markers. So this is the gene here readout for these on the side. So for SMIDE 32 on the right. So they're both significantly increased under hypoxia and pressurized conditions. So that really gives people an opportunity to shorten their workflows. We are looking at other opportunities and how Avatar can influence workflows in central nervous system neurons. We're seeing similar type effects. So this is a general neuronal differentiation protocol. We've seen this in some of the cardiomyocyte work here, where we're seeing enhanced and early expression of cardiac troponin under some of these defined settings, and also looking at opportunities in hepatocyte work. And just quickly to show, this is an Avatar user data slide, an early adopter of these differentiation protocols. What you're looking here at the bottom is their enhanced differentiation markers for FOXA2 and LimX1 transcription factors they're using as early differentiation markers in the workflow. These are both enhanced under the blue line here, which is the Avatar condition versus their standard workflow. And more importantly for them, the markers are obtaining functional cells, functional dopaminergic cells for their Parkinson's disease modeling studies, is enhanced tyrosine hydroxylase expression, and critically aldehyde dehydrogenase expression is enhanced. So we think the Avatar can be used to enhance a number of differentiation protocols, not just motor neurons. So the next thing we want to be able to show now here, where we think you can add the Avatar into a number of steps in your workflows in stem cell biology, be it reprogramming, in early induction, and internal differentiation work. So we encourage you to try that. Now, quickly switching to immunotherapy, this is part of another primary cell type where we feel that there's enhanced benefit by using the Avatar. And so what we think we can offer here, outlined in orange, we have an increased potential in expanding all donor cell types, be it T cells, B cells, and NK cells, and I'll show you some data quickly here on that. We have rapid expansion of both non-activated T cells and memory cells. And we think overall, this really presents the opportunity to help shorten the vein-to-vein time as people are starting to think of the time frame of moving cells from a patient, modifying them, expanding them, and moving back to the patient. So how are we modeling this? This is some data here I'll show you on some PBMC and T cell culture and phenotyping work we've done. We used basically a standard workflow, negative enrichment of T cells in this case, and we also adopted general immune consortium guidelines on which markers to use for these panels. So I think one of the early things that stood out for us that we're really excited about is we tested a range of oxygen and pressure conditions in which to grow these cells. And what we showed on one of the first things here with these 12 donors was enhanced expansion under optimized combination of oxygen and pressure. Now, this enhanced expression immediately provides workflow benefits in terms of more cells over your current workflow if you normally grow cells out to 2 weeks. Or arguably, you can save yourself days here by reaching your threshold number earlier under an Avatar optimized condition. So now we tested a number of conditions, and we want to show you here some data to show you how pressure can influence cells. So this is looking at mean total T cells. So this is CD3 positive cells as they've been grown out over 2 weeks. And our conditions here are really just taking into account pressure as the variable to understand its influence. And what we're showing here is that under normoxic conditions with 5 psi or 2 psi, you have a significant and consistent expansion response in these T cells. Now, what about hypoxic condition? Now, you think of it as a show down here, an arterial environment is really represented by a normoxic state with some force on these cells. So this is think of this as the arterial environment at 15% O2 and 2 psi, whereas under a hypoxic environment, we've defined here as 5% O2 with an additional 2 psi pressure. And you can see that in either under a normoxic situation, as you saw previously, or now under a hypoxic condition, pressure does enhance expansion of the T cells. So what does this really mean for you? When you take this all into consideration, you can see that by leveraging the avatar to create a more normal T cell microenvironment outside the body, you can reduce the time it takes to get relevant, maybe even therapeutically relevant levels of T cells in a significantly shorter amount of time. In this example, we proved that pressure is a critical component to T cell culture, and specifically CD8 positive cells. We want to look at that a bit further now. We can see that when we looked at activation markers by flow cytometry, we can see that we're getting an enhanced expansion of the non-activated cells. So this will include some of the naive cells and memory cells as part of the population. The next thing here, when you look at this per donor, you can see that in this donor pool, some of the cells did not grow out to 3 weeks. They weren't able to consistently grow out to 3 weeks. However, when you add the pressure component, you're able to expand all donors in this population. Similar under hypoxic environment, so this allows you to consider how might my cells be working long-term under a more hypoxic setting that you might find in a compartment of the body. And you can see here too that you're able to understand that, predict that a little bit better under pressure where all the donor cells were able to be expanded. Now, there are some differences in the different subsets to how cells respond or their sort of favorite environment, if you want to think of it that way. So on the left here, we're just showing CD8 cell expansion for those same donors, and you can see that actually 2 psi was sort of the optimum setting for expanding CD8 positive cells. On the right side, we're looking at CD4 positive subset, and you see that 5 psi is sort of the setting that really enhances their expansion. So in this sense, you have the ability to optimize or tune your culture conditions in favor of your cell type of interest. We've also worked on other cell types and profiled those. So these are some data around B cells. For those of you not familiar, B cell present antigens to T cells, so and basically alert them to activation. And you can see that B cells here also prefer a more pressurized setting in terms of optimizing their expansion. We're also undergoing phenotyping of a number of these cell types so we can better understand their functional capacity. Equally, even though this was a culture media not optimized for NK cell work, we were able to actually have NK cell populations thrive under this setting as well. So the avatar really provides the opportunity to increase expansion of not only T cells, but your favorite immune cell type of interest. That technology can help expand difficult donor samples and basically enhance their growth. And cells under the tumor microenvironment, defined here as 1 to 5% oxygen with some pressure, they showed changes in both cytokine release, metabolic markers, and checkpoint receptor expression. So I'll turn this back to James, and he'll speak a little more about some of these markers.
Great, thanks, Bruce. All right, I'll continue by telling you a little bit about primary tumor cultures, mostly from our collaborators, and immunotherapy-focused applications. So before I begin, I just want to profile some of the researchers who've had access to early versions of the Avatar systems, as well as individuals who've actually made the financial commitment to incorporate the Avatar to their daily workflow. Dr. Johan de Bono is a premier prostate cancer research at ICR London. He is interested in using the Avatar system to generate PDX and organoids from prostate tumor samples and for applications such as drug screening and biomarker discovery. Dr. Lawrence Frong is across the street. He's at the Parker Institute for Cancer Immunotherapy at UCSF. He is a pioneer in studying tumor-infiltrating lymphocytes and interest in understanding the contribution of the tumor microenvironment with respect to the metabolic changes of lymphocytes. Dr. Carlos Caldas is at the CRUK in Cambridge. He's a premier breast cancer researcher, is an expert in cancer genomics and PDX and organoid development. As well, I'd like to highlight some of our corporate partners, as well as academic institutions, again, that were early adopters, both from the NCI, Bluebird, Merck, as well as Bayer Healthcare. And so the Avatar tumor workflow is really comprised of kits that is provided by Xcell. There's the tumor dissociation kit, basically allows you to collect fresh biopsies, transport them to your lab, and you have the entire kit available to you to dissociate the tumor tissues apart from the supporting cell type into single cell suspension, and then to propagate these using both substrate and xeno-free chemically defined media cells, media to expand cells of interest. And so if we were to dive into this workflow a little bit further, we have a very gentle tumor dissociation buffer. We often like to say garbage in, garbage out. And so if you have a lot of viable tumor cells to begin with, your chances of a successful culturing exercise is greatly heightened. And so the workflow allows you to, again, dissociate your cells, and that could be both cancer cells or other cancer subtypes like cancer stem cells, as well as interesting subtypes, including infiltrating lymphocytes, could be established in a co-culture model system. And we can start to culture these cells under a variety of different oxygen and pressure conditions to mimic diverse microenvironments from bone, lung, brain, and liver, as well as other tissue types. And what we're able to do is analyze the particular clones or cell types that propagate under these defined oxygen pressure conditions. And we could then perform downstream molecular analysis or omics-based approaches to discover or to understand the different clonal selection properties imbued by the microenvironment in question. And so here I'm going to show you some data from, again, creating organoids or viable tumor samples from patient biopsies, including ascites fluids, apheresis products, as well as from PDX models themselves. The image here on the right is a staining for cytokeratin, which is in green, red, which is EpCAM or epithelial cell adhesion molecule, and blue, which is adaptive for nuclear staining. And this is an image of an organoid. Let me zoom up a little bit. And this organoid was developed and cultured over 2 months. And what we've seen from this sample was that it was collected only from a fine needle aspirate, about a milliliter of solution of heterogeneous cell types. And we were able to, again, get this small amount of cells and actually propagate the tumor cells in question. And so there are other areas of this specific sample where you have intricate tumor intercalation, if you will, with the nearby supporting cell types like fibroblasts, which tend to take over the culture. But what we show here is that the tumor cells actually coexist and are probably synergistic with these supporting cell types, as often we've observed that both the presence of both supporting cell types and tumor cells would likely result in rapid expansion of these tumor cells. And so this is another image, again, depicted from a fine needle aspirate from pancreatic ductal adenocarcinoma, where we show on the left is an image of a developing spheroid that is expanding quite well versus a two-dimensional or well-adhered tumor cells. Again, the markers highlighted in red are epithelial cell adhesion molecule, markers in green is cytokeratin, and blue will be DAPI. And so what we can show is that from the same tumor sample, we get different colonies that appear very, very different. And you can apply both organoid or, again, completely suspended spheroid-based culturing systems or arterial workflows that we have developed internally. So switching some gears, I've shown you that we can culture tumor cells. And I'm going to show you a little bit some data about immune culture and its impact on cell-based therapeutic applications focused on immunotherapy. And this is a simple exercise where we took 12 donor samples and took their PBMCs and basically cultured them under a standard CO2 incubator or artificial environment at 20% O2 and 0 PSI. And we've also cultured them under what we believe to be mimicking the vasculature environment at 5% O2 and 2 PSI. And the tumor microenvironment, which is operating at a lower oxygen content at 1%. And here we show that genes highlighted in green are all increased under 1% O2 and 2 PSI, culturing conditions that mimic the tumor microenvironment. And some of the key markers that folks are looking for, like PD1, ICOS, CTLA4, and LAG3, are all increased under low oxygen and high pressure, mimicking tumor conditions. And again, we've shown this independently. We could reproduce this data from our collaborators. This is unpublished data from the Fallen Lab, from Kiara Rankin, an excellent postdoc who's been evaluating the avatar for almost a year now. And basically what she showed is that on donor PBMCs, cultured under low oxygen pressure, we see that this could lead to significant decrease in interferon gamma, TNF-alpha, and interleukin-10 levels compared to the same PBMCs using the same media cultured under a conventional or standard incubator conditions. At the same time, when we look at their CD4 positive T cells, we see that PD1 levels, again, under a low oxygen microenvironment, increases across over a spectrum of 3 days. And this is PBMCs that have been stimulated with activating beads, CD3 and CD28 beads. At the same time, we also see ICOS is increased. The donor's CD8 T cells are also behave similarly in the fact that PD1, again, is increased across over 3 days in CD8 positive T cells when cultured under tumor hypoxic conditions. ICOS, on the other hand, are increased significantly as well. So what does this tell us? When we put cells, specifically immune cells, in different areas of our bodies, the primary, the vasculature environment, as well as the tumor microenvironment, there's something about the microenvironment that changes the very nature of these cells. And this leads us to sort of the next question that I want to highlight. The idea that we can, again, take parameters such as oxygen and pressure to not only tune the cell, but to direct them into a certain path that is of high interest. And in this case, it is taking an exhausted T cell and then converting that into a more naive, more activated state. And the hypothesis is that a lot of these cell-based products that are being rapidly expanded ex vivo leads to a highly activated or exhausted state that might decrease its therapeutic potency. And so the simple idea is taking cells, providing better culture conditions to provide fitter, more efficacious cellular products. And we have evidence from the literature, and this is, I believe, the first report showing actual animal in vivo data where they took cytotoxic T lymphocytes cultured under hypoxic conditions, and they reinserted these same cells back into tumor-bearing animal models to look at tumor shrinkage and overall survival. And what they found was that hypoxia preconditioned T cells are far more effective at shrinking tumors in mice than T cells that were cultured in a standard CO2 incubator at ambient oxygen conditions. So this leads me to sort of the the final sort of data set that I want to briefly share with you. And it's the simple idea, and you can swap out sort of NK cells here for T cells, but the general idea is we are performing both functional assays, phenotypic assays, looking at marker expression, as well as molecular analysis using next-gen sequencing to determine and to integrate all of this information to determine what is and what constitutes an effective cell-based product. And I think one of the most compelling things about our technology is that we can then start to model this under the context of the dynamic tumor microenvironment. How does my cells change to a change in the microenvironment? And so the NK tumor killing assay workflow we want to highlight is we took donor-derived NK cells that have been enriched using magnetic bead-based separation and expanded under 4 different avatar conditions, all with varying oxygen, both operating under normoxic conditions and low oxygen condition, and varying pressure between low and high pressure levels. And we expanded these cells for about 7 days, 7 to 15 days actually, and then performed co-culturing cell killing assays with prostate cancer cell line D145. And we looked and quantified the cell killing rate using immunofluorescence, flow, as well as an impedance-based cell analysis system. And again, the purpose of this experiment was to determine whether or not avatar cultured NK cells were more effective at killing cancer cells than their conventionally grown NK cells. And just for the conclusion, what we found here is, again, this is an impedance-based cell killing diagram. And what we show here is that the average cell killing rate for NK cells that were grown under standard conditions was markedly lower than the cell killing rates observed by NK cells that were propagated under avatar conditions, highlighted here as AB1 and AB2. And when you look at the remaining number of tumor cells, we find that there are about 30% of tumor cells that are resistant to NK-mediated cell killing under conditions in which NK cells were expanded under standard conditions. However, NK cells expanded in avatar conditions resulted in far fewer tumor cells remaining. And to summarize, what we show here is that under defined avatar conditions, there are NK culture conditions in which it leads to the most therapeutically efficacious cell killing rates. And we show here is that 5% oxygen, 0 PSI, led to only about 10% of the tumor cells remaining. This means that NK cells killed off 90% of the tumor cells. At the same time, under more oxygenated environment at 15% O2 and elevated pressure at 2 PSI, NK cells also showed similar cell killing rates. So this tells us that largely independent of oxygen, we can actually promote NK cell killing of tumor cells just by adding pressure to the culturing conditions. Now, I'd like to just sort of pause there for a moment and let this sink in. A lot of this data is early, and a lot of the advancements that we have made in cell-based therapeutic development is exciting, but we believe there's it's sort of a large opportunity where, again, better culture conditions, a better understanding of cell state could potentially lead to a better clinical outcome. And at Xcell, that is what we're working towards. And I'd like to thank everyone's attention, and I'll be happy to answer questions. Thank you so much.
Excellent. Thank you, James and Bruce. Thank you so much. We do have a couple of questions. So, question, if I remove cells from this microenvironment of the avatar, does the signature degrade? What happens?
So I'll field that one. So the data I've shown you on the any of the stem cell workflows, just as an example here, cells you can typically remove and do your normal cell, your feeding, media exchange, Typically, we don't see any changes in sort of gene expression within the workflow time of 15, 30 minutes. You'd be doing those sort of things. Keep in mind, in these protocols take place over the number of weeks, and these phenotypes we see are dramatic and persistent. We're going through this normal workflow. There's no evidence at this level that there's any influence over the long-term state of the cells. So simply doing your normal cell culture maintenance, you're good.
Excellent, thank you. We have another question about how can you receive a demo, and I can answer that one. You can go to our website at xcellbio.com, so it's xcellbio.com, and in the upper right-hand corner, there's a tab that says Get a Demo. Just click there, give us your information, and we'll set up a demo for you. Okay, that's all the questions, so thank you so much for your attendance. If you have any questions, please feel free to visit the website. Thank you so much, James and Bruce.